Hello everybody and welcome to Seed Banking 101. My name is Aisyah Faruk and I'm from the Millennium Seed Bank Partnership. The contents of this workshop will be delivered by various members of the MSB. It is so great to have a lot of interest for this workshop and we hope that we will find the following afternoon sessions interesting. As it is currently 4 AM where we are, the first 2 hours of this workshop will be pre-recorded, including this presentation. But don't worry, our contact details will be made available throughout the different sessions, so if you do have any burning questions, please do get in touch. Or, if you're lucky enough to bag a place for the live session after the break, you're more than welcome to ask us questions then too, by which time we should be fully caffeinated. So this is how the afternoon is going to look like. We will start with session 1, which will be delivered by members of our UK team. There will be some sections that are very UK specific, but most will be relevant to everyone here. This session will take roughly 28 minutes. The second session will focus on seed drying, processing, and quality assessments. This session will be roughly 45 minutes long. The final of the pre-recorded talks will be in session 3, which is all to do with managing your collections and data, and this will be around 49 minutes long. A word of caution, the final talk will run over the allocated break time, but as these will be made available to you throughout, you're more than welcome to return to it at a more convenient time. Session 4 will be live and will begin with a Q&A, followed by smaller breakout rooms. We've had to limit numbers for this session, so apologies if you weren't able to grab a place, but as I mentioned before, if you have any queries, please do get in touch. Okay, so let's begin with session 1, collecting. Hello, and welcome to the first of our pre-recorded videos. This video will be delivered by members of our MSBP UK team, Jennifer Peach and Stephanie Miles. They will guide you through the process of making a good quality seed collection.
You may know the Millennium Seed Bank as an international partnership, but we also have a long-standing remit to conserve the flora of the UK and currently house over 8,000 collections of native seeds, including the majority of our bankable native species and threatened archaeophytes. So what are we looking for from a conservation seed collection? Well, firstly, the seeds must be collected legally and safely. The correct identification of the species is also essential. The genetic diversity present in the collection should be as representative as possible of the donor population. It also has to be sustainable. That is, that there's no impact to the reproductive potential of a population by removing too much seed. Finally, we need data. A collection is only useful if we have enough information about the wild population and the sampling strategy. So almost all of these points are confirmed prior to harvesting any seed. This video will highlight the steps that you'll need to take prior to making your seed collection. The first and sometimes the most difficult step in making a seed collection is ensuring you have permission from the landowner. We always require landowner permission, and that can be a signed landowner permission form or written or verbal consent. You'll find our landowner consent form in the resources, which is preferably signed and returned to us. If you have any difficulty on obtaining permission, please do get in touch. For SSIs, we have general assent from Natural England, but are required to give notice to the area delivery team to collect on a SSI. You can contact me, Jenny, if you have any queries on how to do this. For Schedule 8, please contact Steph in the first instance, as she is licensed to collect from Schedule 8 taxa and can assign collectors to work under her licence. You'll also need to ensure that you're working under a site-specific risk assessment. If you're not collecting as part of an organisation, we can assist you with any health and safety requirements. If you're unsure, please contact us.
If possible, it's useful to arrange a visit to the site before the day of collection. This is to confirm identification of the target species when flowers are present, and perhaps to familiarise the team with characteristics to identify the plant. Also to ensure there's no potential for confusion with other species or risk of hybridisation. Taking a herbarium specimen at this stage will help to capture key characters such as flowers. It may also reduce the chance of turning up at the site on the day you want to collect to find that the seeds are not ready for collection. If seeds are immature on your pre-collection visit, you can estimate when to fix a collecting date. Also, the site can be assessed in advance for any specific hazards for the risk assessment. Here we have Jenny, who is going through the checklist for all the equipment that we might need for a seed collecting trip. So we've got our herbarium press, we've got a mobile phone for communication, we have various sizes of seed collecting bags and tags. These are cotton and paper to allow air circulation. We've got our water bottles and our biosecurity kit, which is disinfectant so that we can clean our boots if we need to. We've got plant ID, books and maps, a hand lens and our cup testing equipment. And last but not least, we have our first aid kit. So we always ensure that we have plenty of everything. We mustn't forget our risk assessment, and we take plenty of blank data sheets with us. We don't want to run out of those. And last but not least, we have copies of our landowner information sheet, which we can pass to any landowners that we're meeting on site.
With some plants it's obvious when seeds are mature. This dandelion, for example, is just waiting for the seeds to be taken by the wind, and this is a prime example of the ideal collection time for seed banking. A seed at the point of natural dispersal is usually mature enough to withstand the drying and cooling used to conserve. Each plant is different, but there are some general rules to consider when considering seed maturity. For example, colour change, coat hardening, firmness, and dispersing naturally already. Don't be fooled by something like this lodula. It may look like it's turned a nice dark brown, but actually once you've removed the exterior, you'll see that the actual seeds are really still very green underneath. This dandelion might look like it's starting to clock, but actually if you open up the seed head, the seeds are still extremely green and it wouldn't store well in long-term conservation conditions. What we're looking for is something more like these collections you can see here. These broma seeds have been easily removed from the parent plant and they've turned a brown colour. This spigularia can be crumbled out of the capsule easily, and the actual seeds you'll see are very dark brown and firm with a hard seed coat. This scandex is just ready to disperse with very little pressure. Remember to always look with your hand lens and always cut test a seed to know for sure what's going on inside. At this point we know what, where and when to collect, and we have permission to do so. But there are still 3 criteria we need to meet before we go ahead and make this collection. The first is to be sure that the seeds are of sufficient quality and quantity for long-term conservation. We generally aim to collect at least 10,000 seeds, although this is not always feasible. We also need to ensure that the seeds collected are mature and that any quality factors, such as infestation or empty seeds, are taken into account. Second, we need to be sure that the collection is sustainable and won't jeopardise the health of the wild population in any way. And finally, for the collection to be useful, we need to be sure that the collection conserved has captured the genetic diversity of the donor population. To confirm that the collection will meet these criteria, it's important to carry out a population assessment prior to any harvesting. For the purposes of seed collecting, the definition of population is quite general. So we would say a group of individuals capable of interbreeding that occupy a defined geographical area. To ensure we have a collection of sufficient quality and quantity, we first need to estimate how many seeds we have in the population. We're going to look at this in some detail and demonstrate a process that can be followed to make this assessment. Every time you assess, it should become quicker and easier. Remember, the ultimate aim is to make sure there are enough good quality seeds to make a collection. So we'll start by estimating the number of plants with seeds which are at or near natural dispersal. It doesn't have to be exact, but you'll need to walk the site and understand the distribution of plants across the space. If the population is large or spread out, but homogeneously spread, count the number of plants per square metre and multiply out. The number of plants can be multiplied by the number of mature fruits per plant. This doesn't need to be strictly fruits, but some way to make it easier for you to assess, for example, pods, capsules, seed heads per plant. At this point, you'll need to be really clear about your target species morphology. What do the seeds look like? And how many are you expecting to find? Are they in fruits, capsules, pears? Take a sample from different sized and spaced apart plants to get an average. Finally, your total can be multiplied by the number of viable seeds per fruit. As mentioned earlier, it's not possible to know this from looking at the exterior of the seed, so you'll need to perform a cut test. So that is examining the interior of a sample of seeds to assess whether they're full, empty, dead or infested. So the aim is for more than 10,000 seed, but sometimes this is not achievable, especially with smaller populations of threatened species. However, if it's a large population, it sounds like a large number, but this can be fairly easy. Once we have our estimate of the number of seeds in the population, we can then ask about the sustainability of the population. We need to be very careful when collecting seed that we're not causing any damage to the wild population. As a rule, our safe collecting limit is harvesting no more than 20% of seed available on a given day. And we'll take that down to 10% for small populations of threatened species. For high priority collections from fragile populations, it's generally safer to collect smaller amounts over multiple years than to collect more intensively. Thank you for watching, and please get in touch if you have any questions. Video 2 will be covering harvesting seed, data collection and dispatch to the MSB.
Hello everyone, welcome to part 2 of the Collecting Seed series, which will again be delivered by Jennifer Peach and Stephanie Miles from the UK team. In the previous video, you would have an understanding of what to do prior to making a seed collection. In this video, Jen and Steph will take you through what to do on the day of collecting, including data recording, how to treat the seeds immediately after harvest, and how to make a herbarium specimen. At the very end, we will also feature a video on specialist collecting for aquatic species and the things that are needed to be considered when doing them. Thank you.
Once you're ready to start collecting, make sure everyone in the team is familiar with the identification of your target species and take care that there's no confusion with other similar species present. Review the site risks and set a time and place for rendezvous. Use the correct bags to collect into and label your collection with the number, taxon and date. The next step is to decide how the collection will be made.
Grid sampling across the entire population is generally most effective, as illustrated in this diagram. Collectors work in a narrow strip across the extent of the population, so that areas are not sampled twice. Organise the team so that everyone works within sight and earshot of another team member. We want our seed collection to include genes from across the population, so ensure areas of abundance and the edges are included. At least 50 individuals should be sampled, but how many individuals would need to be sampled to achieve 10,000 seeds? The more plants sampled, the better, and as many as time allows. How will we keep to the limit of no more than 20% of the available mature seed? Perhaps sample all the mature seeds from 1 in 5 plants, or 20% from all plants. The data that accompanies a collection makes it more valuable for lots of reasons, but particularly for any future uses of the seed. It's better to have too much information than too little, and record at the time of collection, because otherwise it's easy to forget details. Use a simple numbering system. Numbering systems are important to prevent getting collections mixed up. Usually, collectors' initials with sequential numbering is good. This is the basic data required for a biological record: who, what, where and when. We should be able to refine the population using this data. Record if the taxon is a subspecies or variety, and it's also useful to record who identified the taxon and the confidence level of determination. Habitat data provides information on the conditions in which the plants are growing. It's useful to know whether there are any environmental factors which may be modifying the plant's growth or seed production. For example, you could put grazing by rabbits or sheep in here. This data is referred to if we're propagating plants from the seed in the nursery at Wakehurst. Sample data is the information that informs the capture of genetic diversity from a population, so it's really important to record this. The data gathered during the assessment should be written here. In the plant description box, we record details for the species, including characters used for identification. But only use those that are apparent from the plants that you're looking at. For example, don't copy down text from a flora, as this information will be transferred to the herbarium label if there is a pressed specimen. The key point for post-harvest handling is to keep the seeds or fruits cool and dry to minimise damage. In the field, keep them out of the sun and don't leave them for too long in hot and airless places such as cars or rucksacks. Don't pack too much into the bags so that air can circulate. When back from the field, keep them in a well-ventilated, cool room. If damp, spread the material out in a single layer so that moisture can escape quickly. A herbarium specimen is a pressed, dried, mounted plant specimen. They are curated and housed at Kews Herbarium, which is one of the largest and oldest in the world. While a herbarium specimen is a valuable addition to the seed collection, one should only be taken if the population is large and healthy. They can be used for taxonomic research and naming updates. Tissue can be sampled for DNA analysis, and they are a biological record often used to inform the distribution of a species.
When making a herbarium specimen, we use the same collection number for both the seed collection and the herbarium sheet, so that they can be cross-referenced easily. Write the collection number, the date collected, and the species on the outside of the newspaper and on jewellery tags attached to each specimen. This is in case the paper and the plant get separated by accident. Here we're making a pressed specimen using 2 individuals from the Cardamine hirsuta population. One is at flowering stage and the other contains the fruits or siliqui at varying stages of maturity. As this is a widespread species and an annual plant, we have uprooted the whole plant to include all characteristics. If it's a perennial plant, it is not advised to uproot the whole plant unless you need to show characters in the root or demonstrate that there are rhizomes present. Include as many characteristics as possible in your herbarium specimen, such as flowers, fruits, both attached to show the arrangement on the plant, seeds, which you can put in a small envelope if easier, and include stolons if present. If the plant is too large to fit on a sheet, you can take a part of the plant, but it must show the arrangement of leaves and a section of the stem. Keep your specimen dry. Change the papers regularly, especially if damp when collected, or if the plant is fleshy or contains latex or sap. At the MSB, we dry in low temperature drying ovens, or we keep the specimens in dry rooms. Rapid drying helps to keep characters such as flower colour. It's important to record any features of the plant that will not be shown clearly on the pressed specimen. And these can be listed on the label which is attached to the sheet when filed in the herbarium. For example, the habit, whether it's a tree, shrub or herb, the height of the plant, the colour and texture of any bark, whether any latex or sap is present and the colour of the latex or sap. There may be underground plants that are difficult to include on your sheet, such as a bulb or a corm. Describe the flower colour, as this can alter quite a lot during the drying process. Flower colours can fade during drying. Does the flower have any scent, or are the leaves aromatic, or anything else unusual about the plant? There's a plant description box on the collecting data sheet which can be used for these notes. And all other key information on the collecting data sheet will be converted to the herbarium label, such as collector, date collected, location and habitat notes.
Seeds are produced in a wide range of environments, but whether you're collecting 30 foot up a tree, 2 feet underwater, the general collecting principles still apply. There will, however, be certain challenges to overcome. Steph and I have spent quite a lot of time collecting seeds from aquatic plants. That's both on and in the water. The most notable difference when undertaking this type of collecting is the health and safety requirements. We both have water safety and first aid training, and have developed equipment lists, safety protocols, specifically for aquatic collecting. The risk assessment we use is a dynamic one, which we regularly reassess throughout the day to consider changes in the weather, site safety and personal safety. So for example, hypothermia is a real risk when working in water. Knowing the species is really important with aquatics. Identification can be challenging, and even knowing what the seed looks like. This, for example, is the flower of Rupia shirosa. The seed is at the very end of the very curly peduncle here. It may be difficult to understand the size of a population if much of it is underwater, so collect conservatively to ensure that the collection limits are not exceeded. We want to control the drying of the seeds, so this is one of the few times that we would collect into plastic in order to keep them submerged.
Thanks for watching these videos. We hope you found them helpful, and please get in touch if you have any questions.